In pharmaceutical manufacturing, drying is defined as the precise removal of moisture or solvent from a material to achieve a targeted level of dryness. This process locks in chemical stability, guarantees physical uniformity, and ensures that the finished product meets strict shelf life compliance standards. While a wet granular mass is vulnerable to microbial growth and chemical degradation, the removal of the solvent converts that mass into a robust, free-flowing solid, ready for compression or packaging. Because moisture drives degradation reactions, drying acts as the ultimate bottleneck for product viability. An improperly dried batch results in the chemical breakdown of active ingredients and total product failure. Industrial dryers prevent these failures by balancing two foundational physical mechanisms: heat transfer and mass transfer. Heat transfer is the thermodynamic energy supplied to the material. Heat penetrates the particle, raising internal temperature and vaporizing the trapped liquid solvent. Mass transfer follows, continuously sweeping that newly formed vapor away from the particle's boundary layer. To keep this extraction running efficiently, engineers must strictly control four variables: the system temperature, the airflow velocity, the gas humidity, and the total available surface area. Every piece of commercial drying equipment is simply a specific engineering solution designed to optimize this thermodynamic balance. We can see how these solutions scale by looking at this engineering schematic. The tray dryer represents the simplest method, relying on hot air circulating over a static bed of wet granules spread across horizontal trays. Because the material sits in a static pile, the surface area exposed to the air is restricted. This makes the process slow, limits it to smaller batches, and introduces the risk of uneven moisture removal. Moving to the center of the schematic, the fluid bed dryer offers an engineered upgrade designed for high-volume tablet granule production. Instead of blowing air over a static pile, high-velocity hot air blasts upward through a perforated bottom grate. This forces the dense particles to erupt and suspend in the air, creating a floating cloud that maximizes the exposed surface area of every granule. By maximizing surface area, the fluidization process operates up to 15 times faster than a standard tray dryer. Convective fluidization is highly efficient for robust granules, but the intense kinetic energy and high heat will destroy highly sensitive thermolabile compounds. Materials like soft gelatin capsules or complex antibiotics chemically degrade when exposed to standard drying temperatures. To extract moisture without applying destructive heat, engineers turn to vacuum drying. By reducing the atmospheric pressure inside a sealed chamber, the boiling point of the solvent is artificially lowered. This lower boiling point allows rapid vaporization to occur at significantly safer temperatures. Vacuum drying effectively decouples moisture removal from high heat, saving the chemical structure of delicate active pharmaceutical ingredients. For liquid formulations that must be instantly converted into a dry powder, manufacturers rely on the spray dryer. As detailed in the lower schematic, the liquid feed is pumped through a nozzle using an atomization technique to create fine droplets. When these hit a downward stream of hot gas, the moisture evaporates instantaneously, leaving behind a uniform powder. However, even the brief heat exposure of a spray dryer will decompose vital biological products like blood plasma, viral cultures, and specific vaccines. These complex materials require lyophilization, commonly known as freeze-drying or cryodesiccation. This thermodynamic phase diagram illustrates the process. The material is frozen solid, and the chamber pressure is dropped strictly below the thermodynamic triple point. The material then moves directly from the solid ice region into the gaseous vapor region, bypassing the liquid phase. This direct solid-to-vapor phase change is known as sublimation, and it is driven by maintaining a hard vacuum environment. The critical nature of this technique is evident in biological preservation. This historical vial of dried smallpox vaccine remains viable specifically because it was lyophilized. For biologicals, bypassing the liquid phase through sublimation is the only physical method to preserve complex molecular integrity. Theoretical dryer efficiency is useless without rigorous, continuous parameter monitoring under good manufacturing practice, or GMP conditions. Engineers must be just as careful to prevent overdrying as underdrying. Stripping away essential structural water can trigger a loss of drug potency or cause the solid to physically crumble. Facility operators rely on calibrated sensors to track inlet and outlet temperatures, as well as atmospheric humidity throughout the drying cycle. Drying acts as a strict compliance gateway. A single failure in parameter control yields a suboptimal product and necessitates the mandatory rejection of the entire batch. Pharmaceutical drying is the precise application of thermodynamic control to guarantee physical uniformity, API stability, and strict regulatory compliance. To explore advanced technical resources for GMP drying training, equipment validation, and process optimization, check the links in the description below.